Okay, let's unpack this. We are diving deep into the next evolution of genome editing. For years, the conversation was all about CRISPR-Cas9, which was, let's be honest, revolutionary. But it was also kind of a brute force tool, a molecular scissor.
That's a perfect way to put it,
Yeah.
yeah. If CRISPR was the blunt but game-changing scissor, we are now firmly in the age of surgical instruments.
Mystical instruments.
I mean, tools that can rewrite the genetic code one single letter at a time. We're talking about base editors, or BEs, and prime editors, PEs.
And what's absolutely key here,
Yeah.
the paradigm shift that really defines this new generation of tools, is their whole approach to safety. Traditional CRISPR had a fundamental flaw built right into its mechanism, didn't it?
It did, and that flaw was the double-strand DNA break, the DSB.
Right.
Traditional CRISPR-Cas9 creates that DSB, and when the cell tries to, patch it up, it often uses a process that's really error-prone. It leads to these unpredictable insertions or deletions. We call those indels.
And why is that messy fixing process such a threat? Especially when you're thinking about a drug you have to deliver to a whole person.
Because that DSB is fundamentally toxic to the cell. It's like a giant DNA damage alarm going off.
Wow.
It can trigger severe DNA damage response pathways, like activating the tumor suppressor p53, which can just cause the cell to self-destruct. Or worse, if the cell tries to stitch multiple breaks together, you get these huge chromosomal problems, translocations, rearrangements.
So the new tools, BEs and PEs, they're designed to completely sidestep that.
Exactly. They're engineered specifically to avoid the DSB entirely, and that delivers, well, unprecedented precision and, crucially, what we call editing product purity.
That distinction avoiding the DSB, that really is the difference between a potentially safe therapeutic and one that carries way too much risk. So let's look closer at base editing. If it's not cutting, what is it actually doing to the DNA?
Base editors perform a highly targeted, a very localized chemical modification of a single base.
They're not cutting, just
Yeah.
changing.
Think of it less as surgery and more like molecular dentistry. They just convert one base pair into another. So an adenine base editor, an AB, can convert an AT pair to a GC pair,
Okay.
or a cytosine base editor converts a CG pair to a TA pair. It's changing the letter's identity without ever touching the DNA backbone.
And we already have hard evidence that this approach is much safer, even in really complex settings like making next-gen cell therapies.
We do. When researchers compared adenine base editors to standard Cas9 in allogeneic CAR T-cell manufacturing that's making personalized immune cells, the ABEs were just superior across the board.
How so?
The base editors resulted in far less activation of those DNA damage pathways we talked about and significantly fewer translocations. In fact, those big chromosomal errors, the translocations, were only seen in the Cas9 edited products.
Wow. So avoiding the DSB directly translates to a cleaner, safer final product.
It does.
But even with these safer editors, the field is always trying to reduce any leftover risk. What about off-target effects? How do you make sure the editor isn't accidentally doing its job where it shouldn't?
That's the problem of Cas9 independent deamination. Basically, the base editor protein acting on its own without being perfectly guided. It's a known liability.
So what's the fix?
Well, scientists have gotten extremely clever. They've developed these new BE variants, like YE1, BE4, by just tweaking a few amino acids on the protein. And these newer versions show substantially reduced unintended deamination compared to the older ones.
They're tightening the molecular reins, so to speak.
Exactly. They're making these variants far more promising for any therapeutic use, where minimizing any off-target activity is just non-negotiable.
So base editing is great for fixing those single-letter typos, A to G, C to T. But what if you need to do something more complex? Like what if you need to search for a sequence, snip one strand, and actually insert a whole new piece of code? That gets us to prime editing.
Right. Prime editing, or PE, is what people call the ultimate search and replace function. It can handle insertions, deletions, and all kinds of base substitutions. It's way more versatile.
And it's a more complex machine.
Oh, yeah. It consists of a Cas9 nickase, which only cuts one strand of the DNA, not two, and an engineered reverse transcriptase. And this whole thing is guided by a prime editing guide, RNA, or pegRNA.
And that pegRNA does two jobs.
It does. It acts as both the locator and the template for the new sequence you want to write in.
That sounds like a major leap in flexibility. How does that complex mechanism make it even more precise?
It's the requirement for multiple independent steps to all happen correctly. The editor has to bind, then it has to nick the right strand, and then it has to use the template for reverse transcription.
3 different security checkpoints.
Exactly. Because it requires the alignment of 3 precise molecular events, it dramatically enhances the editing purity and the targeting specificity compared to base editors or, much older methods. It ensures the edit happens exactly where and how it should.
So we have this incredible toolkit of precise, highly engineered editors. Which brings us to the million-dollar question, or maybe the billion-dollar question. How do you actually get this big molecular machine into the right cell, across the cell membrane and into the nucleus, especially in vivo? That's the delivery dilemma.
That is the critical bottleneck for the entire field. The payload, whether it's mRNA that tells the cell how to build the editor, or a pre-made protein complex, RNP, it's huge. It's not just going to drift inside.
So for in-body treatments, we have 2 main options on the table.
Right. We rely primarily on 2 very different methods. The traditional viral workhorse, adeno-associated viruses, or AAVs, and the modern non-viral approach, lipid nanoparticles, LNPs.
AAVs have been the gold standard for gene delivery for decades. What are the trade-offs when we use that viral approach for these newer, bigger editors?
Well, AAVs are great at getting into certain tissues, but they have some clear limits. First, their packaging limit is tiny.
It can't fit the whole thing.
Right. Since base and prime editors are so large, you often need these dual vector strategies where you split the editor into 2 pieces and deliver them separately. That adds a lot of complexity and it just crushes the overall efficiency.
And there's a bigger, maybe more concerning long-term risk. AAVs tend to provide long-term, sometimes almost permanent expression. Having a powerful editor running 204/7 in your cells sounds risky.
It is a profound safety concern. Prolonged, constant expression of any editor, even a really precise one, just increases the window for long-term, maybe unseen off-target edits to build up over time. The longer it's active, the higher the cumulative risk.
And that's exactly where lipid nanoparticles, the LNPs, come into the picture as this game-changing modern alternative.
Absolutely. LNPs are these tiny synthetic particles about 50 to 120 nanometers. Their genius is their chemistry.
What are they made of?
They're usually made of 3 key things. Ionizable lipids that help them get into the cell, structural lipids like cholesterol, and steric lipids like PEG that help stabilize them in the blood. They're basically nanoscale submarines built to protect and deliver a payload.
And this isn't some theoretical technology. Crucially, it's already been proven safe and effective at a global scale.
Precisely. LNPs were first approved to deliver a small interfering RNA drug called Onpattro. And of course, the whole world saw how LNP platforms were used for mRNA delivery during the COVID-19 vaccination efforts. They are manufacturable, stable, and safe.
So if the AAV risk is long-term expression, what is the LNP's killer advantage for gene editing? What does it do that fundamentally shrinks that risk window?
LNPs facilitate transient exposure. This is the single biggest safety advantage.
Transient exposure.
Right. When you deliver the editing agent inside an LNP, it's only active for a very short window, maybe hours to a few days, before it's naturally cleared by the cell. You get the surgical hit you need, and then the tool just disappears.
So it dramatically minimizes that risk of long-term exposure you get with AAVs.
It does. It's a huge difference. The AAV editor factory can run for months, even years. The LNP delivers a one-and-done surgical strike.
That makes perfect sense. It's the difference between doing a quick, precise surgery and, leaving the scalpel inside the patient. Okay, let's move on to seeing these tools in action. Some of the most compelling early successes are in these really complex neurological diseases, which have historically been so hard to treat.
And that's where this high precision really shines, because neurons don't regenerate. You have to get it right.
Right.
So let's look at trinucleotide repeat diseases, or TNRs, like Huntington's and Friedreich's ataxia. These diseases are caused by these unstable DNA repeats. CAG for Huntington's, GAA for Friedreich's, that have this awful tendency to expand over time.
Somatic expansion.
Exactly. And this expansion is particularly bad in affected neurons, and it's what drives the disease forward.
So if the problem is this relentless expansion, the goal must be to just stop it. Jam a wrench in the works.
That's the strategy. Researchers use specific base editors to strategically introduce interruptions into those repeat tracks. For Huntington's, they used a CAG-CBE to convert some of those disease-causing CAGs into CAAs.
And did it work? What were the results of jamming that molecular wrench in there?
It was incredibly successful. This strategy was shown to reduce somatic repeat expansions in both patient cells and in mouse models. The CAG-CBE introduced interruptions in a really high percentage of alleles, somewhere between 39% and 65% across multiple different repeat sites they tested.
Wait, up to 65% correction on a tricky repetitive sequence in the brain?
Yeah.
That's phenomenal.
It's a huge surgical hit rate in a very complex system.
And the Friedreich's ataxia example is even more interesting because it sounds like the base editor is basically mimicking a form of natural protection.
Yes, that's probably the most elegant part. Introducing AAT to GSE interruptions in the GAA repeats for FRDA actually mimics natural genetic variations that we see in people who are either disease-free or have a much later onset of the disease, even if they have long repeat tracks.
So the editor is creating a stabilizing effect that evolution itself has already selected for.
It seems so. It's a huge validation of the whole approach.
Okay, that's base editing. Now let's switch to prime editing's power to fix a single letter mistake in the brain, specifically in this devastating condition, alternating hemiplegia of childhood, AHC.
AHC is a severe childhood brain disease, and it's often caused by these specific point mutations, like D801N and E815K, in a gene called ATP1A3. And correcting a single point mutation deep in the brain is an incredible challenge.
So how do they deliver the prime editor, and what kind of efficiency did they get inside the central nervous system?
They used optimized prime editing strategies, and they delivered it using AAV9, the viral vector, via an injection into the ventricles of the brain in mouse models.
And the results?
When they looked at the efficiency at the transcriptional level, so in the RNA, they achieved exceptionally high on-target correction. In the hippocampus and the cortex, the correction efficiency hit between 73% and 89%.
73 to 89%. In the memory and processing centers of the brain, that's almost miraculous. What does that mean functionally?
Well, that level of correction, of rescuing the transcript, led to a significant rescue of the critical enzyme activity, the 8P1A3 ATPase activity in the tissue from the mice. It wasn't just fixing the letter, it was fixing the function.
And it doesn't stop there. Base editing even showed it could extend lifespan in a disease as aggressive as prion disease.
Yes. The strategy there was to use base editing to introduce a premature stop codon into the PRNP gene. This just shuts down production of the prion protein that drives the disease.
And did it work in a living animal?
It did. When they used highly optimized editors in humanized mouse models, the treatment extended the average lifespan substantially. In one group, it was extended by at least 59% compared to the controls. It's a really powerful proof of concept.
So we've established the incredible precision and efficacy of these tools. But let's circle back to the elephant in the room, safety, and the risk of prolonged exposure from AAV delivery. We have to talk about what happens when these powerful tools are expressed for too long or at too high a dose.
This is a critical lesson the field learned when it pushed the boundaries of where editing could even happen. Researchers used a special base editor to target and correct a pathogenic mutation in the mitochondrial DNA.
The mitochondrial genome, so separate from the main nuclear DNA.
Right, a huge technical achievement. But what happened when they delivered that editor using the long-term AAV system?
What was the catch?
Well, the on-target editing worked, but when they delivered it via AAV9, the researchers saw this really striking dose-dependent toxicity. Mice that got the highest dose, a 10x dose, developed severe consequences.
Like what?
Significantly lower body weight, clear signs of muscle atrophy, and just an overt mitochondrial dysfunction phenotype. They got sick.
And the survival rate. That tells the real story.
Alarmingly, 44% of that high-dose group died before the end of the study. And the toxicity was specifically linked to dose-dependent mtDNA off-target edits. The editor was hitting the wrong spots in the mitochondrial genome.
This is the perfect, if sobering, illustration of the delivery dilemma. The editor itself was precise enough to work, but the delivery system, the AAV, kept it running for too long and at too high a concentration.
Exactly. The tool didn't fail, the exposure window failed, and while they didn't see an increase in off-targets in the main nuclear DNA, the mitochondrial genome, where the editor was highly active, just suffered from that prolonged exposure.
It's an urgent reminder of why transient delivery systems like LNPs are so important.
You need the editor to get in, do its job, and then get out. Quickly.
So what does this all mean for the roadmap ahead? We've achieved this unprecedented surgical precision. We've seen success in the brain, the eye. We've seen editing rates up to 80% for diseases like Stargardt, and we can even edit the mitochondrial genome.
The precision is here. The proof of concept for treating diseases that were once completely untreatable is undeniable. But that mitochondrial study is a huge cautionary tale. Prolonged expression of these potent editors is a major safety risk, and that reality is pushing the entire industry toward non-viral transient delivery methods like LNPs.
The goal now is fewer, more flexible tools delivered as safely as possible.
Right. The goal is to refine these systems so you need fewer compositions of matter to benefit the largest number of patients.
So considering how remarkably successful these targeted, precise editors are in all these preclinical models, across the most challenging diseases you can imagine, what do you think will be the next major bottleneck in translating these cures to patients? Will it be refining that tissue-specific delivery? Getting those LNPs to only hit the liver or only the neurons? Or will it be the sheer logistics of scaling up safe manufacturing to meet the global demand for these incredibly complex synthetic medicines? That's something for you to think about as this field continues its dizzying acceleration.